It's my pleasure to introduce Shyam Prabhakar, our speaker for today's Center for Human Variation seminar. Shyam is a senior group leader at the Genome Institute of Singapore and is also the associate director of the Spatial and Single Cell Systems, also at the Genome Institute of Singapore. For the past 20 years, Shyam's lab has focused on regulatory genomics and transcriptomics, but during the last 10 years, his lab has focused entirely on doing research on spatial and single cell analysis. His lab develops algorithms for analyzing chromatin profiling and single cell and spatial omics. He introduced the concept of histone acetylome wide association studies, where um, um, uh, histone association between autism and TB cases and controls are compared. He leads the Asian Immune Diversity Atlas, the flagship program of the Human Cell Atlas Project, Asia, that is generating the first single cell atlas of Asian immune cells. Sham also holds the multi-tissue tissue map IAF-PP program to identify diagnostic and targetable markers of fibrosis-related cancers and chronic diseases. Shyam trained as an engineer and physicist in Stanford and thus has a solid foundation in statistics, mathematical modeling, and data analytics. I was in Shyam's lab before I joined Broad, and when I joined his lab, I transitioned from doing mathematical biology to genetics and genomics. And in his lab, I learned a lot about statistics and mathematical and data analytics. And what I really appreciate from learning from him is doing rigorous and statistical analysis to really separate signal from noise from your data. And also, what's interesting is that Nirmala, who is also a Brody now, was also from his lab, and the three of us worked on a project together where we identified and characterized the first primate-specific neurodevelopmental enhancer. Today, Sham will talk about his recent work on two spatial algorithms and his analysis of Asian Immune Atlas. We are looking forward to hearing about your recent work, Sham.
Thanks so much, Rick. Amazing to be here. And yeah, two of the stars from my lab are here, Rick and Nirmala. Spent some great years working with them and becoming good friends. Uh, yeah, I'm sure they've already done great stuff here. Um, my daughter is also supposed to join. We'll see. We'll see whether she really. Ah, yes, there she is. Uh, okay, let's get started. Two different topics, but we connected by one thread, which is that how we're influenced by our surroundings, human beings influenced by their environment, and cells influenced by the cells they see around them, right? But the concept is the same, and I think we want to study all of these interactions using single cell and spatial technologies. There's a lot of slides, but it's okay. I don't mind just finishing the hour without going through all the slides. I'd rather just have questions and have a conversation on whatever I'm showing. So please feel free to interrupt. You don't have to wait till the very end because I'll be going through many topics. If you wait till the end, it's become very discontinuous. So I don't need to tell this audience single cell is very popular for a long time and because tissues are diverse, right? That's why you need to analyze them one cell at a time. And that takes us straight to the Human Cell Atlas, perhaps the largest international single cell consortium. And single cell and the goals of the atlas are to create a comprehensive reference map, and I underline the words comprehensive reference map of human cells. Okay. What does comprehensive mean? At one level, it means every cell type in the human body, but I'll argue that's not the only kind of comprehensive we want. And here's how comprehensive it is in terms of, say, continental ancestry, continental geography, origin. Not very diverse, right? Even most of the unknowns are also, the y-axis is, I guess, number of cells. Yeah. In PBMC datasets in CZI cell by gene, which is not exactly the same as the Human Cell Atlas, but it overlaps a lot. And you can see here that it's overwhelmingly European ancestry or European ethnicity, whatever you call it. Very little of other stuff, right? So it's not, the HCA is currently not so comprehensive in this sense, right? And why is that a big deal, right? So because human diversity really matters for health. It's not merely a curiosity, right? So you have population-specific disease risk variants that you will never understand unless you study specific population. And if you don't study them, you don't characterize those risk variants, then you will misdiagnose people, right? So basically, there's a fault of one size fits all medical practice, right? It has to be, it doesn't, it works mostly, but it's going to fail catastrophically in some cases, right? And then there's, how you respond to drugs depends on your ancestry, and it's different between males and females as well. So there's known, aspects of human diversity that have medical clinical consequences. And that's why, and even geographically, right, disease incidence is diverse across the world. And some parts of the world, there's some endemic parasitic or infectious diseases. In other parts of the world, there's endemic metabolic disease and so on because of some lifestyle genetics interaction. So if we really want biology that leads to medicine, that biology has to be done across the world in diverse environments, in diverse populations, diverse genetics, right? And that's why we need a diverse atlas. We keep saying HCA is a healthy reference, but there's no one healthy reference. You need a healthy reference for each population group in each part of the world, right? And if we don't, we will definitely have healthcare disparities. Now, human geneticists are very familiar with polygenic risk scores not porting to other ancestral, other population structures, other ancestries. But it's not just that, even your gene expression-based diagnostics may not transfer very well to other parts of the world. So that's where the genetic diversity network of the Human Cell Atlas comes in. I hope I've, at least got you thinking along the lines that diversity is important in these single cell studies. And not just that it's important, because it's so understudied, right? It's a scientific frontier. It's an opportunity. It's not merely an obligation for us. It's an opportunity to do some really cutting edge science in a field that's wide open. So what is Human Cell Atlas Asia doing? So HCA Asia is the Asia arm of Human Cell Atlas. And I would say very important to the diversity goals because 60% of the world's population lives in Asia. And that's a lot of people. And it's extremely diverse. The two most populous countries in the world are in Asia. And tremendous, as you can see from this map and just imagine, extraordinary amount of environmental, socioeconomic, of course, ethnic, cultural, genetic diversity, right? Going from, what you would call, say, European ancestry up here, South Asian ancestry, Southeast Asian, lots of admixture, and the Middle East is incredibly genetically diverse. And of course, you can see how the climate changes across all of this, right? So Human Cell Atlas Asia has a big job to do. And about 5 years ago, I think, we started out on the Asian Immune Diversity Atlas. As our first step in that direction, we said, okay, we can sample blood in a lot of places. Let's collect PBMCs from a bunch of countries in Asia with harmonized protocols and study how human diversity affects our molecules and affects our cells, right? And build a healthy baseline for precision medicine in all of these countries and all of these populations. And then, of course, in Asia and many parts of the world, it's not enough to have a healthy reference. You want to immediately compare to some matched disease cohort. In our case, we're looking at diabetes and cancer. I won't show the diabetes results yet, very immature. I'll just show the healthy reference. But yeah, this is the overall scope of IDA. Where are we today on the healthy side? We have diabetes donors profile also, but on the healthy side, we have 619 healthy Asian donors from 6 ethnicities, 5 Asian countries, Japan, South Korea, India, Thailand, Singapore. at the moment. And then the next wave of countries in IDA are just starting up. But these countries have already generated data, and that's what I'll show you today. You can see here that there is a lot of, I'm coloring these dots. Each dot is an individual. This is a genotype PCA plot to show the genetic diversity of the IDA samples. And you can see here the substantial genetic diversity here, spanning a reasonably broad range of ages. And this is what we're really excited about. So the size of the circle, each circle is one data set in cell by gene, one PBMC data set in cell by gene. The size of the circle is how many donors you have, I think. Yeah, or how many cells you have, I don't remember. Yeah, I guess donors. And then each slice of the pie is one self-reported ethnicity. Okay. And you can see here that the other pies are, smaller, or one pie is almost equal size, but they don't have the well-categorized diversity that IDA has, right? And that's really important. Some of these cohorts may actually be quite, environmentally, ethnically, and ancestrally diverse. You just don't know it because they have not collected that information or they have not reported that information, okay? And if you don't collect it and you don't report it, the fact that your cohort is diverse does not help you so much. It gives you a nice population average, so that's valuable. It's better than not having a diverse cohort. But you lose your ability to characterize the consequences of that diversity, right? And that's why I would just make a very strong point that metadata is important in any of these, you can say, single-cell epidemiology studies. And the other thing that's important is you really want to harmonize your protocol. So there's two ways you can go. One is you can centralize all your sample collection and sample processing, which is hard if you're collecting samples in many countries. You can fly your team over there and have them collect samples there. Then it'll be really, and bring them back and then process them centrally. Then it'll be as harmonized as humanly possible. But of course, one goal of IDA and of many parts of the Human Cell Atlas is to support local science, right? And so it's a push-pull. There's no magic answer for how you manage the trade-off between those two. In IDA, we went with mostly collecting samples and generating single-cell libraries in those respective countries, okay? And certainly in some parts of IDA, that has really helped them get their own grants, their own research grants from the national funding agencies. And that is the capacity building we're looking for, right? If we had just pulled the samples, brought it ourselves, it would not have helped them in any way to nucleate capacity there. And so we're very proud of that. But the downside of that is you get huge, I shouldn't say huge, but you get batch effects, okay? And we try to minimize, what I mean by batch effects. If you collect blood in Thailand, you collect blood in Seoul, you collect blood in Yokohama. You can collect it slightly differently, you can process slightly differently. And then you think that you're looking at differences between Japan and Thailand. You're not, you're looking at differences between how those samples are processed in Japan and Thailand, right? And that's the batch effect you want to avoid. So we put all our protocols on protocols.io. We harmonized how the samples were collected, how they were processed, and then we centralized the library generation was distributed. The data, the sequencing was mostly centralized, and low-level data analysis was centralized. And then we pass the annotated files or annotated objects to all the respective countries, and then they can do their own science, right? So we did a lot to reduce batch effects across countries. It's not perfect. They still exist. But as you'll see, we still managed to learn a lot. So in a sense, it was a journey. How do you learn about human diversity in a study that's distributed across countries? So here's one example that immediately jumped out at us, okay? Because we expected, so Japan and Korea are genetically very similar. And if you look at them in genotype PCA space, there's almost no difference compared to the distance between, say, Japan and India or China or something like that, right? But we saw that Tregs are drastically, almost twofold depleted in the Korean population. So this is telling us something, presumably about environment. Okay? And we did the control. We also profiled Lonza European control samples in all these countries. Tregs are not depleted in the Lonza European samples profiled in Korea. Okay, so we suspect that this is real, and it's maybe genetic, but perhaps more likely some environmental effect, right? That makes this population distinctive. As you can imagine, lower Treg proportion in your blood could predispose you to a number of inflammatory or autoimmune conditions. So we try to look up, does Korea have a greater incidence of autoimmune disease, for example? And we couldn't answer that question because we couldn't find any studies where they have systematically used the same identical criteria across Asian countries to characterize these disease incidence or disease prevalence, right? But this is kind of surprising, and maybe this kind of finding will inspire. Let me see if I can get the mouse there. No. Yeah? Where is it? Ah, there it is. Yeah. Well, maybe this kind of finding will inspire epidemiological studies of disease incidence in these countries. What else can you do across countries? So we can look at male-female differences, and there you have internal control, okay? Even if you have batch effects across countries, those batch effects presumably affect the male and female samples approximately equally. And so the male-female delta is still reliable and robust to those batch effects, right? So what is the male-female delta and say naive B cell proportion in the blood? Say among Chinese and ethnicity Singaporeans, among Indian ethnicity Singaporeans, and so on. You can see among people of Japanese ethnicity, there's almost no male-female difference in IB cell proportion. But in Indian ethnicity and Malay ethnicity in Singapore, there's a massive difference, and this is log to the base 10, okay? So 0.3 difference in log to the base 10 scale is a twofold difference in naive B cell proportion between males and females of Indian ethnicity in Singapore, almost no difference between males and females in Japan, right? And this is another way of visualizing the same thing. So when you say that you're studying human diversity and you're studying male-female differences, be careful. You're studying male-female differences in that population. It may not be true of male-female differences in other populations, right? And that was the surprising aspect of this here. So ancestry or ethnicity, you can say, confounds all of these other variables. And then age effect, I'm calling it ancestry, but it's really ethnicity, okay, what we studied here, because it's self-reported ethnicity. Age effect, you can see that CD4 naive T cells go down as you grow older from about 20 years of age to 70 years of age. And this is the average across all Asian populations. But if you look at individual Asian populations, there's tremendous variation. And in Chinese ancestry or ethnicity Singaporeans, it actually goes slightly up, right? So again, you cannot, even the most basic statements you make about your blood cell types, you can only say that they are true of your population in your country. You can't say that this is human, these are human trends, right? And that was a surprising thing. More or less everywhere we looked, we saw profound modulation by ethnicity and ancestry. And here's a single cell. I'm showing single cell data, but I'm showing you things that you can, in principle, learn by facts also. So here's what you can't learn by facts, okay? So this is a single cell UMAP. Each dot is a cell, but the dots are so small and so many dots that they're merging into these islands and continents in gene expression space, right? So location of each cell in this gene expression space represents its phenotype or its state at some level. And so these are the T cells. So naive CD4 T cells. Oh, off again. Yeah. Naive CD4 T cells, naive CD8 T cells, NK cells, CD14 myeloid, CD16 myeloid, dendritic cells, and so on. Okay. So the blue and the orange here, the orange means that you have more in this plot. Orange means that females have more of this cell state than males, and blue a lot more, meaning twofold or fourfold more. And blue means that males have more of this cell state than females. Okay, again, fourfold more easily. And then you can see Indian Singaporeans versus Chinese Singaporeans. You have profound, let's say naive CD4 T cells, okay? These are all naive CD4 T cells by the textbook. If I did a FACS analysis, I would say Indian and Chinese Singaporeans have the same number of naive CD4 T cells. But if you look at it in single cell state space, there's an axis here. There's this axis. This is the T cell differentiation axis in this direction. Transverse to that is the male or whatever, Indian Chinese differentiation axis, right? Which shows up only when you have single cell data. Now, so I hope I've convinced you that cell phenotypes are profoundly influenced by all these aspects of our diversity. So how much is it really, right? And sex is kind of the benchmark. We all accept as biologists that there are sex differences in our cells, in our transcriptomes, gene expression levels. What we're seeing is that, I'm saying ancestry here, it should be ethnicity, okay? Ethnicity explains as much variability in cell type proportions as sex, right? And this is just among Asians, okay? So Asians are presumably more similar to each other than they are to, say, people of European ancestry or African ancestry living in Europe or Africa or North America, right? So if we did either across the whole globe, it's quite conceivable that ethnicity would actually explain more variability in our blood than made the male-female difference, which is striking, right? When you do an epidemiological study, you always say, okay, I want a balanced cohort. I want males and females in my cohort. You don't often say, it's happening increasingly, you don't often say, I want people of all ethnic backgrounds, all ancestries in my cohort. But the data are telling you that that latter is just as important, right? At least for blood. And I know Kristen has done a lot of work on this in GTEx and shown that the answer is actually different in different tissues. So that is also a really important point to make, that you want to do this kind of study in a whole bunch of tissues. Now, we started with blood because blood is easier. And easier to standardize also, not just easier to collect. So does it mean anything in terms of disease susceptibility, right? Now, the world is accustomed to looking at genetics and saying, okay, this means something for your disease risk. This has biological significance. So GWAS is kind of the benchmark or cornerstone of a lot of biological inference in the human genome, right? So here's UTS2. It's an EQTL SNP of UTS2 is associated with basal metabolic rate, body fat percentage, diastolic BP, and so a bunch of metabolic phenotypes. But that EQTL SNP explains just a very small amount of variation in UTS2 expression. But presumably that 50% or so variation in UTS2 gene expression is enough. You know, that SNP causes about 50% variation in UTS2 expression. That's enough for that SNP to cause differences, observable differences in your metabolic phenotype, right? Now, 50% expression difference, compare that to what we're seeing here between Singapore Indians and Singapore Chinese, okay? So, and this is log 2 scale, a difference of 3. You're seeing an 8-fold difference in UTS2 expression between Singapore Indians and Singapore Chinese, and each dot is one cell type, so across all cell types. So if a small difference in UTS2 expression measurably affects your basal metabolic rate, etc., imagine what an 8-fold difference in UTS2 expression will do, right? And this is why we really have to characterize expression states and cell phenotypes, because you'll see variation that's often much bigger than what GWAS explains. But we assume reflexively, based on tradition in the field, that the GWAS SNP is important, but we ignore that there's much bigger baseline gene expression differences between populations, right? That's, in my view, even more important in terms of effect on phenotype. Here's another gene, FCR1A. It has a SNP, EQTL SNP, that has a, reproducibly associated with allergies. And FCR1A expression is about, so this is log to the base 10, and it's about it's at least threefold higher in Singapore Indians than Singapore Chinese or Singapore Malays, right? And that's again a bigger gene expression swing than that GWAS SNP produces. So clearly, I believe that all of these studies, um, at least single cell studies, single cell epidemiology, can move towards disease risk prediction, just like we use polygenic risk scores, right? And okay, it's the same thing. We've already shown that. So what have we learned? So cell proportions are variable in the blood. Asians are not all the same. You'll see a lot of studies that do characterize diversity in their cohorts, but they characterize it at the continental level, right? There are studies that look at Asians, Europeans, Africans, say in the US, Asian ancestry. Uh, but what our results are showing, are results showing that among Asians, so Asians are not a monolith. Among Asians, you have tremendous diversity. So you need to characterize people's ethnicity or ancestry at the subcontinental level. And then you will have more precise baselines. And when you average across all Asians, you're blurring out a lot of meaningful variation. Male-female differences depend on ancestry and ethnicity. Effect of age depends on your ancestry and ethnicity. And effect of ancestry or ethnicity is about as strong, within a factor of 2, of the effect of sex, even within Asia, right? So I made this point repeatedly here. I think, and okay, one last point I want to make here is I'm deliberately using ethnicity. Some of these ancestries need to be changed to ethnicities because what we're seeing is not necessarily the effect of the genotype. It's not that people, Chinese Singaporeans and Indian Singaporeans have different DNA, and that's why they look different. They also have different diet, uh, different on average socioeconomic status, right? Different social status. And many other aspects of lifestyle and habits are different. And that could be what we're seeing. So the beauty and the challenge of transcriptomics is that it gives you some additive combination of your genotype and your environmental influences. It's hard to disentangle those two, but that's our job. But you do want a readout that reflects both of them, right? You don't want to look at genetics alone. All right. IDA is a very large team. I want to highlight Kian Hong, who was a postdoc in my group, who's been driving the Singapore side of the IDA project, and a bunch of other collaborators as well. Jay Shin, Wong Yang Park, and I nucleated IDA 5 years ago or 6 years ago at an HCA Asia meeting. And lots of collaborators have come in. We have annual IDA workshops at HCA Asia meetings, very large team. Nirmala was spearheaded, was at the birth of IDA and helped to organize the whole consortium right at the beginning, sort out some of the protocols. I think the protocols were based on her PBMC single cell RNA-seq work, so really a pioneer in this area. That's the IDA part. Maybe I'll stop here to take some questions before getting into the spatial section.
I was curious if you have enough subjects who might have some genetic admixture, but were, let's say, Japanese people who were in Korea and collected in the Korean cohort, that you could start to disentangle this difference between genetics and environment. And then a follow-up question is, how much metadata do you have about these people? Could you start to at least hypothesize something about what this
Great question. That's precisely what Wong Yang Park, who's leading the Korean cohort in IDA, is trying to do. He's trying to recruit people of Japanese ancestry and Chinese ancestry in Korea. Not easy. He's not there yet. But you brought up, there is tremendous genetic and ethnic diversity within each country. So the Thai team in IDA has collected PBMCs from 4 different ethnicities within Thailand. And that'll be fascinating, and won't be surprised at all if we see profound differences in there. Disentangling genetics and environment
is really the key. A lot of Asian countries are ethnically and genetically fairly homogeneous, but there are some exceptions. So Singapore is a major exception. So we're very lucky that in Singapore, a lot of the results I showed are Chinese, Malay, Indian ethnicity in one country, collected side by side, processed in the same lab, batch randomized, and so on. So those are the differences that we are most confident about, and they are very strong as well. But the Korean thing, we really would love to validate, as you were suggesting. Yeah. Rakeli?
So you said that the batches that you have for sample collection and processing is confounded with your main interest, which is the diversity between the different locations. And obviously you can look within location or country and look at like sex differences and stuff like that. And I think this is a problem that is becoming all of our like a problem of many because the data sets are becoming bigger and bigger. And I wonder, how do you try to deal with that? Besides looking within data set, do you like do some kind of validation, or do you look within different batches in the same setting? I'm curious.
Thanks so much. Picking up on things that are there in the manuscript but didn't have time to put in the slides. How do you wrestle with the fact that uh, ancestry and batch are confounded? For example, say between Thailand and Japan, right? Uh, One is harmonizing protocols, and that certainly helps. If we did not harmonize the protocols, I think we wouldn't even have been able to make any conclusions at the level of cell subtype proportion. Even that would have been a suspect, right? And then, like you said, we do internal controls. So male versus female, age, and so on. Uh, for EQTL analyses, we regress out both batch and ancestry. We treat them as confounders. So we're looking at shared EQTLs across countries. But then we found that, we also did a quick analysis of, say, Malay ethnicity EQTLs in Singapore. And we found some new EQTLs that have, very strong in the Malay population. There's virtually zero EQTL signal in the other populations in either. So there again, we believe it because when you do an EQTL analysis, you're comparing the A allele to the G allele in the same population. So you have internal controls. So it's robust, right? So there's a lot you can do. But then you mentioned validation. That's really the gold standard. So again, for some Chinese, Malay, Indian differences in Singapore, we were lucky to have access to the FACS data from the Singapore Longitudinal Aging Study, which is a very old study. And there's beautiful correlation between the cell type proportions we inferred from single cell and IDA and the cell type proportion we saw in the Singapore Longitudinal Aging Study by FACS. And some of our conclusions about cell abundance differences in Singapore were valid. I think all the major conclusions that we reported also seen in the longitudinal aging study FACS data. So that gives us some confidence that we're not seeing artifacts, but of course we need more validation, we need FACS validation in all of these countries. For example, the depletion of Tregs in Korea, we don't have FACS data yet. So all of that, there's a chicken and egg problem in diversity studies. People don't appreciate how much diversity is there, and so they won't do the work that's needed to collect the samples to characterize it. Right. So I think this can hopefully kickstart that process, and then people will start doing the collecting samples for FACS as well, and doing Luminex assays and doing all of the other assays that are independent of single cell RNA-seq to validate this. Kristen?
I was given a microphone, so I'm going to use it. Shyam, do you want to speak a little bit about the extra challenge? Obviously, the goal of doing these is it can be really rich in helping us to interpret the effects of environment. But as you and I know in the diversity group and HCA, the real problem here that we haven't really discussed is the metadata, and it can be very sparse and very different. Do you want to just discuss the whole issues that are associated with getting sort of unified metadata?
Yeah, thanks for bringing that up. We just, in the HCA Diversity Task Force, we just formalized our diversity metadata recommendations and sent it to the Equity Working Group for ratification. most studies are collecting, very diversity metadata is collected very unevenly. So we're trying to propose some standards for basic aspects of diversity metadata, which is geography, ancestry, ethnicity, uh, phenotypic sex, uh, genotypic sex, and some aspects of environment. Although environment is very hard to capture, it's really the hardest part. So in terms of environment, we're using geography as one proxy for environment, but you don't really learn what is it about that geography that makes the blood this way or that way, right? So you can guess about climate from geography, but then you have to standardize that as some variables to correlate with. Um, we collect smoking. We're recommending that HCA collect smoking data. Um, and there was a proposal. So diet is a big environmental influence. There was a proposal to collect that, but it's just hard to standardize a diet questionnaire across the world. So yeah, I don't know if we have the answers. What we propose is some kind of common denominator, minimal metadata that people could collect across HCA so that they can at least do the kinds of analyses I'm showing here. And I didn't show smoking, but actually we've already incorporated smoking into these analyses. It explains some variation in cell proportion gene expression, not as much as ethnicity or sex, surprisingly. But we're not looking at lung. So that could be very different. All these conclusions apply to blood, not generically. Going beyond that, uh, I think we need to say what environmental metadata can we and should we collect, and which of those metadata fields really affects our cells. Even there, it depends so much on the tissue, right? So if it's gut, it's one thing, one kind of metadata. If it's lung, it's another kind of metadata. If it's skin, it's another kind of metadata. And so that makes it hard to standardize. My guess is that all of the HCA bionetworks are also proposing their own metadata specs. So these are the generic diversity metadata specs for all tissues. So the lung bionetwork will collect more detailed information about smoking. The gut bionetwork will presumably collect more detailed information about diet. And then hopefully we can encapsulate environment. But yeah, I'm very much open to suggestion. The field is wide open. Okay, let's get to spatial. Oh, there's one more question.
We have a question on Zoom really quick. I was curious about the sex-based differences in different ethnicities. Were you expecting that or just happened to observe that sex was the largest predictor of within-group differences for some of the ethnicities?
I think we expected sex to have a major impact, but I would say because there's so few studies, single cell studies of this, we did not know quite what to expect, how sex would stack up compared to some of the other variables. And we did not expect at all that the effect of sex would be different in different ancestries or different ethnicities. And I think that's why I say this is a scientific frontier, right? You just at first pass, things fall into your lap that are completely unexpected. Okay. How much time do I have? So maybe another 15 minutes? Yeah. All right, so single cell is not the ultimate technology. Obviously, spatial tells you how the pieces are put together and how the pieces of the machine interact with each other. And that's what we really want to understand in a tissue, right? So, of course, for spatial omics, now in 2024, data generation is relatively easy. If you have a lot of money, of course, then you can do it, right? And you can robustly generate beautiful data. Data analysis is really the bottleneck. Okay? And there's two basic data analysis questions that come up immediately when you generate spatial omics data. How to define cell types, which is cluster cells by their transcriptome, and some other phenotype, and how to identify tissue domains, like say the dentate gyrus of the hippocampus. Okay? So that's an example of a tissue domain. An anatomical region. And a tissue domain could have multiple cell types, but it is still has a coherent function and a coherent cell type composition and architecture, right? How do you identify these two things from spatial data? Spatial data should be better than single cell data at identifying cell types, though paradoxically, the dogma in the field nowadays is that you use single cell RNA-seq to define cell types, then you use spatial omics to tell you where those cell types are in the tissue. Maybe I can convince you in the next 15 minutes that that is the wrong paradigm. Even with the really limited spatial omics data sets we were able to generate last year, before all the technologies improved, we found novel cell states from our spatial omics data that we were never evident in single cell RNA-seq data, right? And now that spatial omics has gotten better, I would say spatial omics is the way to define cell types, not single cell, really. Or you can say they are equally powerful. And maybe next year, spatial omics will be more powerful than single cell at identifying cell types or cell states. Right. So here's a common benchmark in the field, identifying cell types in StarMap mouse visual cortex brain data here. Each color is a cell type annotated by the authors, okay, of that paper. So you can see here that you can see roughly some vertical stripes. These are the cortical layers, but each vertical stripe has multiple colors, multiple cell types, right? So you want to color the cell types accurately. That's problem one. Problem two is you want to identify the vertical stripes, the cortical layers in the brain, because each of them has some characteristic property of cell type composition, right? Both of these problems from the same data set, there are algorithms that solve this. There are algorithms that solve this. We want one algorithm that solves both problems using spatial data. And that's what Banksy is. Banksy is basically, you can think of it like Surat or ScanPy clustering of cells, except, this purple matrix is the Surat or ScanPy gene expression matrix, okay? Each column is a cell, each row is a gene, and then you just cluster cells, these columns, by how similar their gene expression vectors are, right? So I'm off the screen again. This is the transcriptome of one cell that you normally cluster in single cell data. Now, what Banksy does is it appends the average transcriptome of the neighbors. So suddenly your transcriptome is twice as long. So, and each gene appears twice, once for own expression, once for neighbor expression. And then this vector encodes the gradient or the slope, the spatial gradient of the expression of that gene. Right, so each gene now appears 3 times in Banksy. Once the cell's own expression, once the average neighborhood around that cell, average expression around that cell, and third is the slope of the expression around that cell, right? So now you encoded the spatial transcriptome in these linear vectors, and now just throw them at Surat or ScanPy and ask them to cluster as though it's a single cell transcriptome. And of course, you add some weights over here. So you have one free parameter lambda that determines the relative weights of the own expression, neighbor expression, and neighbor gradient components of your augmented feature vector. And and that's all it is. It's fairly elementary. As you can imagine, when you do this, you get a very scalable algorithm. One problem with a lot of existing algorithms in the field is that they run out of steam after 50,000 or 100,000 cells. Right? But one data set, one single tissue slice can give you 1 million cells. So an algorithm that can cluster 100,000 cells can't handle even one tissue slice, right? And scalability is a huge problem in this field. But Banksy is inherently scalable because it's as scalable as single cell RNA-seq clustering, okay? So let's benchmark it. Let's see if it makes sense. So here's a benchmark of mouse cerebellum slide-seq data. And you can see here that this is from the RCTD paper, okay? So this is non-spatial clustering where you can to some extent see these anatomical structures, the granular layer in mouse slide-seq V2, slide-seq V1. But what the RCTD authors claimed was if you use reference single cell transcriptomes, if you have additional information, you already know the transcriptomes of these cell types, then you can deconvolve and much more cleanly, where did the mouse go? Okay, never mind. You can much more cleanly, if you see the second row there, you can much more cleanly identify these anatomical structures. But what Banksy does is it does not need independent single-cell transcriptome information, and I hope you'll believe that it resolves these anatomical structures very cleanly by using the spatial neighborhood expression and spatial neighborhood gradient. And then you don't need additional information to interpret this data, right? It does as well, at least visually, as RCTD. Here's another benchmark, the famous Moffett et al. Mouse brain Murfish dataset. The authors identified this yellow oligodendrocyte cell type because they use non-spatial clustering. They did not use the spatial information to cluster the cells. We use Banksy, and so Banksy found these two clusters, the subcluster 2, subcluster 1, the maroonish cluster and the orangish cluster. So basically two subtypes of oligodendrocytes that are spatially segregated, and they have subtly different gene expression, okay? And that's why no one saw them before. But subtly different gene expression of cell states can become a very clear distinction once you have spatial information, right? And that's what Banksy is able to pull out. We could not find any published papers that identified these two subtypes of oligodendrocytes. So we said, okay, can we go back to publish single-cell oligodendrocyte, single-cell RNA-seq data and confirm that these cell types are real using these markers? Indeed, we were able to do that. So the maroon subtype and the orange subtype separated out even in single-cell RNA-seq data once we knew which markers to look at, right? So it is definitely a difference in cell state and perhaps related to the functional differences in these two anatomical regions. So here again is an example where this dogma is wrong. This dogma that single-cell RNA-seq tells you the cell types is wrong. In principle, it does, but there are 10,000 genes in single-cell RNA-seq. You don't know which genes to look at to find that difference in cell state. And so, there are only about 8 genes or 10 genes that differentiate these two cell states. And there are 5,000 genes or 3,000 genes that you use for clustering. So those 8 genes, the signal from those 8 genes gets swamped by the noise from the other, the 2,992 genes. And that's why you don't see it in normal single-cell RNA-seq clustering. Here's another benchmark. Again, I showed you the star map visual cortex. How does Banksy find spatial domains or cortical layers versus the other algorithms? And you can see here that Banksy has a best concordance score, ARI, compared to the ground truth of 0.72 compared to the other methods. And here's a slightly offbeat on the right-hand side, slightly offbeat use of Banksy. So we found that Banksy was finding these clusters in mouse brain organoid data that were at the core of the organoid, and that was actually the necrotic core. So it was not a biological cluster, it was a technical cluster, which is not apparent. But because the necrotic core is spatially coherent, Banksy found it very easily. And we verified with these marker genes, these heat shock protein markers, are markers of necrosis, right? So now we also use Banksy by surprise for QC of single spatial data. And this is just one example. In some spatial omics data sets, Banksy pulls out clusters that are neat squares. And those are single fields of view in your spatial omics data that were out of focus. And Banksy zooms in on that and says there's something peculiar about this field of view, and then you just kick it out in your QC. I mentioned scalability. Here's the numbers. You can see a Banksy scales easily to 2 million cells on a fairly regular computer. So you can analyze your large data sets, and the other existing methods don't scale so well. They take much longer to run, and they give up. They run out of memory after some time. Right? And Banksy can be scaled even more. As Charette scales to bigger data sets, ScanPy and Harmony scale to bigger data sets, Banksy will as well. It's not just us benchmarking Banksy and saying it's good. It's already also been independently benchmarked by Mats Nielsen, whom some of you know from his work developing the spatial transcriptomics technology that eventually made it into 10x genomics. And they recommend in their benchmarking that Banksy has they recommend Banksy as the clustering algorithm to use for 10x genomics Xenium data. Similarly, another group, Yuan Wang's group, published a paper benchmarking Banksy against a bunch of other spatial clustering methods. And again, they said that Banksy correlates best with these manually annotated spatial domains. So yeah, we're very excited that we have external validation. Banksy uses a biologically motivated feature augmentation strategy, right? And what is that? A cell is influenced by its neighbors, right? A cell sees the signaling environment of its neighbor. So you have to characterize a cell not just by itself, but by its neighborhood composition. And it's one algorithm with just one parameter lambda that you adjust. It's fairly robust. You don't have to play around a lot with Banksy parameters. You just adjust lambda and you can switch it between cell type clustering and tissue domain segmentation. And we applied it to virtually every single spatial omics data type, including multiplex protein imaging, and works quite robustly. And it finds new cell types that single cell could not find, right? And it's very fast. So a bunch of labs collaborated with this, and we also collaborated with a private company for this work. But I should particularly thank the people in blue, Vipul and Nigel, and Joseph Lee also down below, who were the developers of the Banksy algorithm. And we applied Banksy and the spatial omics profiling to colorectal cancer, large 30 patients, even more now. We did multiplex FISH, a homemade version of MERFISH plus Xenium on colorectal cancer, which we've been working on for a long time. I'm going to skip over most of this, except to say, again, we found cell states that were not visible in single-cell RNA-seq data. So here you see the red cells. This is a colon tumor. In the H&E, the purplish mass is the malignant cells, and the pinkish rivers between the purplish masses are the stromal rivers, so they contain mesenchymal cells and immune cells. Okay. Now you can see here the red subtype and the orange subtype of the epithelial cells. They are spatially distinct. The orange subtype sits on the edges of the tumor nests, edges of the epithelial mass. And the red subtype sits more towards the necrotic core of the tumor nest, right? So you see each purplish mass has some orange cells on its edges and some red cells further inside. And that's an example of spatially segregated cell states, which spatial omics are very good at catching. Then when we go back to the single-cell RNA-seq data, use these markers that we found from MERFISH and Xenium, then we see that, aha, there is a distinction even in single-cell RNA-seq. We just never noticed it because we did not have spatial location as a guide, right? And here, then spatial omics can also tell you co-location of different cell types, and we're seeing fascinating differences in where these various subtypes are located in the tumor. Some infiltrate the tumor, some are confined. It's like an army that's being repelled, right? So there's neutrophils forming a wall around the tumor margin, but something is preventing them from going in, for example, or certain kinds of macrophages forming a wall. It's like this army invading, but then right at the walls of the fortress, something stops it, right? And you can see those phenotypes very clearly in the spatial omics data. Here's another example of what you get from spatial. And I mentioned you want to see the cell-cell interactions, right? Here's a beautiful example of how just by eye, without using any algorithms, we see that so the yellowish, reddish cells are the mesenchymal cells that are next to the tumor, okay? And they are secreting TGF-beta. The purple cells are the cancer cells and the malignant cells. And the green cells are the malignant cells that respond to TGF-beta signaling by expressing a TGF-beta response marker. And you can see here that the green TGF-beta responding cells are right next to the red TGF-beta producing cells, right? The cancer cells that are far from the red cells are purple. They don't express this TGF-beta response marker. So there's and this we found in two independent data sets. The left is a MERFISH data set. The right with the blue margins is a MERSCOPE data set for colon cancer from WisGen. In both cases, we see this unique TGF-beta responding cell type just at the margins of the tumor nest, right? And another cool phenotype we saw, and yeah, that was merfish mainly. We also generated xenium data over 4 million cells and find beautiful cell types in colon cancer. Here's another example of something, a cell state in colon tumors we found only with spatial data. The orange marks, if I remember correctly, EpCAM. Okay, so the orange cells are the cancer cells up above. And you can see here we've zoomed into a region just at the tumor margin. You see orange on the left and just a bunch of gray DAPI on the right. So that's right at the edge of the tumor, okay, where the tumor is invading into the surrounding tissue. How does the tumor invade? By budding, by forming these small fragments. You see the yellow arrows highlight the tumor buds, these small fragments, these small seeds that the tumor plants and sends out into the surrounding tissue. What is the phenotype of those invading cells? And those are the cells that allow the tumor to push into the surrounding region and grow. And those are the dangerous cells, right? So beautifully in the spatial omics data, those invading cells, they have a different, now down below, the pink and blue plot or image. There the cells are colored by their Banksy cluster IDs. You can see here that the invading cells, the tumor buds, are mostly colored blue. So they have a distinct epithelial cell state. But intriguingly, even you can see the lowest black arrow points towards something that a pathologist would never call a tumor bud. It's a fragment of a tumor nest, and it's also blue. So pathologists say that you have to have 1 to 4 cells isolated, cancer cells isolated, and that's a tumor bud. But what we're seeing transcriptomically that you should probably use a different definition. You should allow up to 20 cells. A 20 cell fragment is still a tumor bud because it has the same transcriptome and it's still pushing into the tissue. And in fact, some of these nests, I'll try really hard to get the mouse. Yeah, here you go. Some of these tumor nests, right? They are kind of half normal phenotype and half the budding phenotype you see here, right? So this tumor nest right at the edge of the tumor has the budding phenotype, even though no pathologist would call it a tumor bud. But it expresses these unique markers. And if we can target, so the tumor budding is prognostic and it's a risk marker. Instead, we could, but that's just eyeballing H&E plots, right? Instead, if we can use the molecular markers of these tumor buds, that's much more precise in prognosticating about outcomes for these cancer patients. And hopefully this will lead us to discovering new targetable. So if we can use drugs that target the markers of these cells, then in principle, potentially, we can halt the growth of these tumors. We can confine the tumor to where it is. Right? So, all right. And I'll jump to one big program we're launching, which is Tissue Map. We just got funded. We're going to do this kind of spatial omics profiling on 2,000 patients from 5 cancers and 2 non-cancer fibrotic and inflammatory phenotypes, combined with H&E data from a much larger cohort of 100,000 to 100,000 patients and matched clinical data for the larger cohort. So with this trimodal data set, we hope to identify this kind of TGF-beta response signaling interactions and correlate those signaling interactions in tumors to outcomes and drug response to stratify patients and identify new druggable targets. So this figure here shows that, 70% of existing drugs target either ligands or receptors. Okay, so we find new ligands and ligand receptor interactions in spatial omics data that correlate with outcome. Those are potentially new drug targets. And that's the goal, main goal of Tissue Map. Large team, mostly for the spatial cancer work. I'd like to thank Libo and Grace for that. And I'm out of time, so I'll just quickly say one thing about this and stop, which is spatial omics gives you cell location, cell transcriptome, but it also gives you cell shape. And most people are throwing away that cell shape information. We came up with an algorithm to cluster cells by cell shape. So these are examples of of cell shapes over here. On the left, you can see the oblong oval shape, the round shape, the heart-shaped cell, and so on. There's a way to, and the main problem with clustering cells by cell shape, by encoding cell shape, is that if I take an oval cell, I rotate it like this, it's still an oval cell. But all existing machine learning and deep learning, so ResNet, if you use ResNet to encode images, ResNet will give a different encoding to this cell and this cell, even though shape-wise it's the same cell, right? So how do you encode shape in a manner that's invariant to rotation? And we came up, you cannot do it fully. We came up with a heuristic for it. It works really well. And once you do that, we can beat ResNet and we can beat any other existing cell shape clustering method. Here's the benchmarking. So MEDOC is our algorithm. It's the top one. It has the tightest cell shape clusters. And then you get these beautiful pictures like this. This is not a gene expression UMAP. Each dot is a cell. This is a cell shape UMAP. Okay, cells are located in this two-dimensional space based on the cell shape. This at this far right end, these are the roundish cells. These are the roundish cells. These are the more irregular heart-shaped cells, two different kinds of irregular heart-shaped cells. And then we color it by this KLF transcription factor expression. You can see here that KLF is highly expressed in all the non-round cells, lowly expressed in the round cells. And there's quite a few genes that we've characterized this way. And these genes turn out to be markers of myeloid differentiation. And when myeloid cells differentiate, they do go from a roundish to a more irregular shape. So you could say this is a positive control, except we're looking in chronic myelomonocytic leukemia, where cell shape is prognostic for risk. And nobody knew, as far as we could tell, nobody knew that these CMML bone marrow cells recapitulate the cell shape differentiation hierarchy of normal bone marrow, right? And so now these cell shapes that are prognostic in CMML, we can assign molecular markers to them and say, okay, why don't you try these markers and have a more objective or more reproducible way of stratifying risk for these patients. So that's just one example. I think cell shape is relevant for fibroblasts, obviously, a whole bunch of cell types. And so ultimately for spatial omics data, you want to look at not just expression and location, but also morphology. And this is a collaboration, Tang Hien and Ong Sing Tiong in Duke. Thank you.
Any questions in the audience?
Hi. I was just wondering about Banksy. So you have the cell's own expression, the spatial neighborhood expression, and then you've also got the gradient. What does the gradient matrix have? Because I guess intuitively it wasn't immediately obvious how that is, it sort of improves it obviously does, but how it improves the resolution.
Great question. So the neighborhood average smooths out edges. So you lose resolution. So the gradient sharply tells you where the edge lies. And that helps you compensate for the smoothing of the neighborhood. Chloe? I think, sure. Chloe was asking, why do we see these cell types? Why do we fail to see some of these cell types in the single cell data? Is it because dissociation somehow killed them, or they failed to dissociate? Or maybe we're not looking at the correct markers, or you absolutely need neighborhood information to identify them. Some combination of all of these. The big advantage of Banksy spatial data is the neighborhood gives you something to correlate with. So okay, the main problem with single cell RNA-seq data is you're always clustering based on 3,000 genes. And all of these cell states are subtle. There are like 5 to 10 genes really that define that cell state. The remaining 3,000 genes look identical, almost identical. So there's almost no way you will see it in the single cell data because 5 to 10 genes are always swamped by 3,000 genes. But if you're able to correlate your gene expression with spatial location, then those things jump out very cleanly. And then PCA, what does PCA do? PCA identifies those correlations, right? And that's why we see it in the spatial data. But the dissociation also is a very good point. Our TGF-beta responsive cell that is bordering the tumor nest, we kept looking for it in the single cell RNA-seq data. We don't believe we see it very clearly in the single cell RNA-seq data. And we have looked at the largest colorectal cancer single cell RNA-seq data sets in the world, including our own. We believe it's because colon tumors are very hard to dissociate. They're quite fibrotic. And so those cells and these epithelial cells, they die en masse when you dissociate. So we're getting some non-representative selection of tumor cells in our single cell data. And that is a big problem for single cell of primary solid tissues. The stuff is lost in dissociation.
Although clearly you heard my question if I shout. I just want a quick follow-up question because you mentioned you've looked at the largest single cell suspensions collective data set for CRC. So how are you approaching integrating your insights from these single cell suspension data sets with your spatial? I imagine you don't want to ignore everything that has been built. So are you leveraging it to kind of define key features to look spatially? And as we're entering a new era where we can look at so many features spatially, what's the value of the single cell suspension data sets, in your opinion?
Great question. Well, spatial data are still at the level of quality where single cell data tells us some things new that are new. So they both, there's a Venn diagram. Single cell is telling us things we don't see in spatial. Spatial is telling us things we don't see in single cell. So they're both valuable today. Maybe next year, only spatial will be useful. Yeah. But so what we do for CRC is we've reclustered all the available single cell RNA-seq data for colon cancer to define cancer cell subtypes. And we've clustered our spatial data to define subtypes. And we try to see what's shared across our spatial and single cell data. And that's what we can analyze integratively. What's not shared, we try to validate externally. So essentially, we take our insights as some superset of what we're getting from single cell and what we're getting from spatial. And they're both useful today.
Hi, a really good talk. Along the lines of sort of fishing out the genes that are spatially correlated amongst like the full transcriptome, have you tried or what do you think about the idea of like running spatial autocorrelation within a cluster of cells to sort of like tease apart those patterns and then sort of like potentially like sort of cross-validate those with sort of non-spatial clustering and sort of help with the interpretability of these sort of neighborhood-driven clusters?
So you're suggesting calculating the spatial autocorrelation from gene to gene?
Basically of genes within a cell type. So like if you do spatial autocorrelation just across the tissue, it's going to be, of course, driven by where the cell types are located. So, but then if you sort of go like within that cell type and then look for spatial autocorrelation within that, then maybe that would help sort of like complement some of the analysis that's from spatial.
As a way to tease apart subtypes, I guess you're saying.
Yeah, or spatial patterns within their, within a cluster, like sort of all things being equal within this cluster, let's sort of reanalyze that and sort of hopefully tease that spatial component apart. Just curious.
Yeah, that's an excellent idea. I think that autocorrelation-based methods are very powerful for feature selection and identifying patterns. So that's in a way a clustering agnostic way of identifying spatial patterns in the data. So I would say we need to combine both. And if you identify for example, the spatial DE algorithm, which was a pioneer in that, looking at spatial variation of gene expression. And then there have been many other algorithms after that based on, geospatial analysis or autocorrelation, cross-correlation methods from geospatial analysis. We are hoping to develop some algorithms in that space as well.
Hi, Shyam. Thank you for the very cool talk. I was wondering, it's quite interesting to see your results on the last part about the morphology or the cell shape identification or using that to cluster the cells. I wonder if you see subtle differences for those cells that like have different show some differences in the sectioning when you slice the when you do the cross sectioning for the slide. And if I wonder if you could use or integrate the expression data to recover or even extrapolate the 3D shape of the cells.
So right now, it's a great question about 3D shape. All the shapes I've been talking about right now are 2D shapes. So obviously inadequate. And what could imagine that the 2D shapes that we're looking at here, for example, like the top branch and the left branch of the shape you map, you could imagine that they're really the same 3D shape, just looked at this way or looked at this way, right, depending on the sectioning plane. Uh, it's quite possible that that's true. It's quite possible that those two arms are really the same shape. We will know only when we have 3D representation of the shape. In principle, we do, because we take multiple z-planes, and we take enough z-planes, you're getting all of, a good fraction of the cells. And this Medoc algorithm can be generalized. It's based on, 1D Fourier transform. So it can be generalized to two-dimensional Fourier transforms uh, to align, arbitrary rotations, align 3D cell shapes, right? We're not there yet. I think already we can get a lot of insights from this. But the main problem is computational. Already all these technologies are doing multiple z-planes and in principle giving you the 3D shape. Algorithmically analyzing that is just going to break your IT, at the moment. Yeah.
We have a few questions from the audience online. You've mentioned that gene expression-based diagnostics may not translate well between different ancestries. There are now foundation models trying to learn gene-to-gene relationships that are specific to cell types. Do you think there is no one cell type specific gene-to-gene relationship, but there are cell type and ancestry-specific gene-to-gene relationships? And do you think the gene regulatory network of a cell type differs between different ancestries too?
That's a great question. And it really relates to how big is the ancestry or ethnicity influence. Um, you can. So I don't have the plot here. I'll try to conjure it up in words. So you can imagine, say, two stick figures representing two human beings, okay? One stick figure is the African ancestry human being living in Africa, and one stick figure is the, let's say, Chinese ancestry human being living in Singapore. They both have heads, arms, legs, right? But if you put them in some gene expression space, those two stick figures won't coincide. So the arms, legs, and head of the Chinese Singaporean individual will be at a slightly different location in feature space. But then you can use batch effect, batch correction algorithms or data integration algorithms to superimpose them so that the head of the Singaporean coincides with the head of the African and the arms of the Singaporean coincides with the arms of the African and so on. That's what your data integration algorithm does, right? And so the arms are cell type A, right arm is cell type A and left arm is cell type B, and the head is the progenitor cell or something like that. And so then you will find some coherent progenitor cell and differentiated lineage A and differentiated lineage B molecular traits. Definitely. It's not like we are different species. We are the same species, right? So you will find cell types, you'll find cell subtypes that are shared across all humans. It's just that the proportions of those cell types will change from human to human, and the precise transcriptome of that cell type will change from human to human. So we're not trying to negate the concept of shared human biology at all. That would be absurd. We're just saying that the differences are big enough that, once you characterize cell types, once you've defined the cell types, you want to characterize how different are they in different ancestries. Yeah, same for gene-gene relationships. There are shared gene-gene relationships across all human beings, but they will be modulated by environment and genetics and so on. And they're modulated strongly enough that some of them will have clinical consequences for diagnostics, for risk stratification, for disease physiology, disease processes, and so on.
Another question online is, in the 10x Visium platform, where one spot is more than one cell type, how would Banksy deconvolute each spot for the cell types?
Great question. So the older generation 10x Visium technologies has about 50 micron or 100 micron spots. And so that covers like 2 to 10 cells. So you're, in essence, you're getting a spatial pseudobulk transcriptome at each spot. RCTD is a deconvolution-based method, and so it needs the single-cell transcriptomes. Banksy does not try to deconvolute. It just tries to cluster the pseudobulk transcriptomes. And it actually does very well. So it's the best performer on the human dorsolateral prefrontal cortex Visium benchmark, where the the people who published the paper annotated the cortical layers in that data set manually. And then the task for the algorithm is to cluster the Visium spots into those layers accurately to match the manual annotation. And without any deconvolution, just looking at the pseudobulk transcriptomes, Banksy outperforms the other methods at delineating those pseudobulk transcriptomes. Because each pseudobulk transcriptome encodes some property of the cell type composition of that layer, as well as a geographical location, anatomical location of that layer. And that's enough to separate out the layers without deconvolution. Right. So we actually don't use deconvolution much. The problem with deconvolution, going back to algorithms like CyberSort, which is like a legendary algorithm, deconvolution algorithm for bulk RNA-seq data, is that it's a bit unstable. You have some reference transcriptomes that you use to deconvolve and infer cell type proportions. If you have some technical bias in your bulk assay, let's say Visium has a very different technical bias from some other assays, your inferred cell type proportions can change dramatically. And some cell types can disappear altogether. You know they're there, but CyberSort or whatever deconvolution algorithm says is just even not there in your data set because of the technical bias in the data, right? So we actually try to avoid deconvolution. And then the beauty of spatial omics technologies is that now they've become high res. Visium HD is subcellular resolution. So we don't need to sweat it out with deconvolution anymore. We have cellular resolution data, right?
Any other questions in the audience? All right, well, thank you, Shyam, for a wonderful talk, and thank you everyone for coming.
Thank you.